Hello dear students. Today we are going to study about a very important topic from heredity. Heredity, which is having maximum weightage in your board examination also. Today we will learn about heredity from the very basic. First of all, the word heredity. Heredity is termed to be transfer of characters from one generation to other. As you all must have heard also that we are getting some characteristics from our father, we are getting some characteristics from our mother. So this transfer of characteristics from one generation to another is called to be heredity. And study of heredity in biology comes under the branch called genetics. So we have to understand one more term, that is genetics. It is branch of biology where we study about heredity. So today we are going to discuss about heredity in the branch of biology called genetics. When we say we are getting characteristics from one generation to another, one generation in which there are many species, in which there are many organisms, and we all know every organism's basic body unit is a cell. So here you can see the picture of a cell, and the cell is controlled by a central unit which is called nucleus. The controller of every cell is called nucleus. Inside the nucleus, there is a thread-like structure. In this, the thread-like structure is called chromatin. This thread-like structure, at the time of cell division, get condensed in form of chromosomes. These structures are called chromosomes. Basically, these chromosomes are transferred from one generation to another during reproduction. Either it is sexual reproduction or asexual reproduction. But these chromosomes are actually composed of the basic unit, the supercoil unit, which is known as DNA. You must all have heard about the genetic material in the cell. This genetic material in the cell is called DNA, or in some viruses it is also RNA. Basically, this DNA stands for deoxyribonucleic acid. Nucleic acid because it is inside the nucleus, and deoxyribose is showing its chemical composition. It is having ribose sugar which is not having any hydroxyl group on the second carbon. So deoxyribose sugar, which is composed with some nitrogen bases and phosphate group and called as DNA, deoxyribose nucleic acid. So heredity, which is transfer of characters from one generation to another. In every cell, all the characteristics are controlled by nucleus. In the nucleus, there is the chromatin thread. Genetic material containing structure is called chromatin thread. This chromatin thread get condensed in form of chromosomes. These chromosomes are composed of DNA and proteins. DNA's full form is deoxyribose nucleic acid. So whenever there is transfer of characters from one generation to another, actually DNA is carrying information from one generation to another, or we say from parents to the offsprings. Now, let us focus on this DNA. which is inside the chromosomes. Whenever a cell divides, as you can see here, the cell is dividing. Whenever a cell divides into daughter cell, its genetic material needs to be duplicated. It needs to be doubled. And that is the basis of heredity, that is the basis of reproduction too. Whenever any cell has to divide, whenever any generation has to produce the next generation, its genetic material needs to be duplicated. So we call it as DNA copying, or we call it DNA duplication. Right? So we will mention it as an important point. DNA copying is a method by which genetic material doubles and transfers from one generation to another. So this is called DNA copying. DNA copying is a very important topic, students, because whenever you are going to learn about heredity, you should have that concept in your mind that whenever characteristics are transferred, always the basic is DNA copying. DNA needs to duplicate itself so that it will be equally distributed in the next generation. It will be equally distributed in the daughter cell. So whenever cell division takes place, DNA copying occur, and DNA copying is required. Why? What is the significance of DNA copying? It is required for continuity of life. Please remember this thing. Continuity of life is the significance of DNA copying. Without DNA copying, life could not continue, because finally the genetic material will get vanished. It will not be transferred to the next generation. Whenever we need that transfer of character must take place, DNA copying is the basic step. Okay. So when we are talking about genetics, students, we have some basic terminologies which we should know, so that we can talk, or we can learn, we can understand about the experiments done in this field. Whenever genetics terminologies are called, first word that we must know are the traits, or we also call them characteristics, or the features which are transferred. The characteristics of the or the features which are transferred, they are called traits. Then the factors. Factors are actually now known as genes. Genes are the functional unit of DNA. We say na that DNA copying is required whenever cell is going to divide. DNA copying is required when transmission of characters are required to be done in one generation to another. This is all because of genes. Genes are the functional unit of DNA which are actually going to function, right? And initially these genes were known as factors. Then we have a term called alleles. Every gene exists in pair, and these pair are in contrasting form. So the contrasting form of any gene are called allele. Like if I am talking about a pea plant, that pea plant is having a contrasting trait. It can be tall, it can be dwarf. So I must say that the gene which is responsible for the height of the plant is having two contrasting form. Either it can be tall or it can be dwarf. So contrasting form of any gene, any trait is called allele. For example, tallness and dwarfness. They are the contrasting form of height. The characteristic is height. So they must be considered to be the alleles of which trait? Height trait. Then we have a term called dominant, dominating, which will not allow anyone else to express itself. So dominant form which always express itself. Like in a family or in the classroom, there are some students who are always dominating, always speak out. They will never give chance to others to speak. So they are called to be the dominating personalities. Similarly, those characteristics which always express themselves, they are called to be the dominant traits, and contrasting of that is recessive. They are always suppressed. In presence of dominant, they will never speak. They will never express themselves. But when they are alone, when they are free, they are not having any pressure, they will express themselves. Recessive alleles only express in absence of dominant alleles. Otherwise, in presence of dominant alleles or dominant traits, recessive traits never express themselves, right? So, recessive alleles are which are not expressed in presence of dominant alleles. Okay, so these are some important terminologies that we must know before going for the experiments in genetics. We must know what are traits. Traits are the characteristics. We must know factors. Factors are the genes which are responsible for transferring of traits. Each gene exists in its contrasting form. That contrasting form are known as alleles. There are two contrasting forms. One is called to be the dominant form, another is called to be the recessive form. After that, few more terminologies are there. Like we have a term called progeny. Progeny means next generation. Very simple. Progeny means next generation. It is also called to be filial generation. Right? So, parent generation will give rise to the offspring, or the parent generation will give rise to progeny, or we say parent generation will give rise to filial generation. All are meaning same. They are called to be the next generation. Then we have a term called pure line or homozygous. There are pure line traits, there are homozygous traits. In this you will mention that pure line traits characters are where both alleles of a gene are similar. When both the alleles are similar, we call it homozygous. Like if I say pure tall plant, right? So I will mention it as capital T, capital T. So both the alleles are same. If I say dwarf, it will be represented by small t, small t. Again, both the alleles are same. It is again homozygous. So when in any genotype, in any characteristics, both the alleles are exactly similar, we call them pure line trait or the homozygous trait. Homo means similar, and opposite of that is heterozygous trait. Hetero means different. Hetero means different. Like heterozygous tall. If I say heterozygous tall, then I will present it with capital T, small t. Both the alleles are different. If I say heterozygous violet flower, so I will present it with capital V and small v, because here both the alleles are different. This is called heterozygous. Then we have the phenotype and the genotype. Phenotype is the physical appearance of any trait. How it is appearing physically, how it is observed, that is called phenotype. And when we represent any character symbol or symbolically, we give symbol to any character, then we are giving its genotype. As I say, tall, I can represent with capital TT or I can represent with capital T small t. So this is known as the genotype. Symbolic representation of any trait is called genotype of that trait. So these are the important terminologies. We have to understand about progeny. What is progeny? The next generation. Pure line, where both the alleles are similar. Heterozygous, where both the alleles are different from one another. Then we have phenotype, where physical appearance of any trait is talked about. Genotype, when we are giving symbolic symbols to any trait, that is called the genotype of the trait. Right. Now we come to the experiments we study in genetics. Whenever we talk about genetics, there is the name of a scientist which is Mendel, and its name is G.J. Mendel, or you will write it John Grieger. It is John Grigor Mendel. He is known as father of genetics because he was the first person who worked in the field of genetics. And the interesting fact is that he was not a biologist. He was actually a mathematician, and he was very fond of gardening. So during his free time, he started doing some experiment in his garden area. He selected a plant which was garden pea plant, Pisum sativum, and he started calculating some results from that on the basis of mathematics formulas, right? And on that basis, he concluded some laws of genetics. Interesting, na? So John Grigor Mendel, J.G. Mendel, is known as father of genetics, and he worked on pea plant. Which pea plant? Garden pea plant. Garden pea plant. We all must remember the scientific name of this garden pea plant, and scientific name of this garden pea plant is Pisum sativum. Okay? Now the question must arise in your mind that why he selected only garden pea plant. Garden pea plant is not available for 12 months. Even it is not so attractive. There are so many colorful plants in the garden area. Then why only pea plant he selected? There were some specific reasons behind that. And the reasons why he selected garden pea plant, they are: number one, this plant is having short lifespan. Okay, in 2-3 months he will get the result quickly. Next, this plant, pea plant, was easy to maintain. That's why he selected the pea plant. Then in this pea plant, it shows both self and cross pollination. Pollination means, I hope you remember, transfer of pollen grains. So transfer of pollen grains may be within a same flower, self-pollination. Or if the pollen grains is going from one flower to another flower of another plant, that is called to be the cross pollination. In the pea plant, both type of pollination were present. Okay. So this plant was showing both self and cross pollination. And last, in pea plant, contrasting, opposite traits, characters were easily available. So these are the reason why Mendel selected the pea plant, not the other colorful plants in his garden. This is very important, why he selected pea plant. After that, Mendel, after selecting the garden pea plant, he find out some specific contrasting opposite traits, characters, with which he will continue his experiment. So those characteristics are also important for your board exams. There were 7 characteristics. One was related with the seed. It was seed shape. As you can see in the picture, the seed shape may be round or it may be wrinkled. So the first contrasting trait Mendel studied in the seed shape was either it will be round or it will be wrinkled. Again in the seed he find out the color. The color of the seed was either yellow or it were green, right? So these are the two traits related with seed. Either the seed shape was round or wrinkled, or the seed color was yellow or green. Here also remember which one is dominant and which one is recessive. In the seed shape, round is dominant, so we will present it with capital R. Wrinkled is recessive. So wrinkled trait is always symbolizing with the same alphabet what we have used for dominant one. If we are representing dominant round trait with capital R, then we have to represent the wrinkled trait with small r. Got it? If the color of the seed yellow was dominant, it will be represented by capital Y. And the recessive trait in the seed color green will be represented by small y, not small g. It will be represented with the same alphabet but in small letter form. Got it? Then there was another trait, third trait. It was the flower color. The flower color were either white or they were violet. Here, violet were dominant, capital V, and white was recessive, small v. Then pod, fruit of the pea plant. The pod shape were either full or it was constricted. So full was dominant, capital F. Constricted was recessive, small f. Color of the pod, yellow was the dominant color and green, here green was the dominant color. Green was the dominant color and yellow was the recessive color in the pod. Then there was position of the flower. Flower position was either axial, axial was dominant, capital A, or the flower position was terminal, it will be represented by small a. And last, either the plants were long or we call them tall, represented by capital T, or either the plant was short, dwarf, and dwarf will be represented by small t. So there were 7 contrasting traits selected by Mendel for his experiment. Okay, what were the 7 contrasting traits? Number 1, in height, it was tall or dwarf. Then flower color, violet, dominant, white, recessive, right? Seed color, yellow dominant, green recessive. Seed shape, round dominant and wrinkled recessive. In pod shape, dominant was full, fully filled. Pod was dominant, constricted was recessive. Color, green was dominant, capital G. Green was dominant in pod and yellow was recessive. So 7 contrasting traits Mendel studied for his experiment. So on the basis of these traits, Mendel conducted his experiment which were based on hybridization actually. We also call them cross because it was including the cross-pollination technique. In this cross-pollination technique, Mendel selected pure line plants. I told you earlier, pure line means homozygous. Let's take an example. The first case, Mendel selected height, 1 character, right? So monohybrid cross is actually, mono means one character studied at a time. That is called as monohybrid cross. So if Mendel talked that I will continue my experiment with one character, that is height, he selected parent plants, the male parent plant and the female parent plant. The pea plant was bisexual, so he selected pollen grains from one plant and he transferred them, dusted them on the pistil of female plant. Suppose the male plant was tall and the female plant was dwarf. And what type of tall and dwarf? Homozygous. So tall and dwarf are the phenotype of this trait. And if I write the genotype, I have to write the homozygous form. So it will be capital TT and it will be small tt. Homozygous forms both. What I have written here? Genotype. So Mendel selected the parent generation. In the parent generation, he selected the pure line tall and pure line dwarf plant and he crossed them. Whenever crossing is done, always the parent plant will form gametes. Because of meiosis, in the gametes, the chromosomes become half. Gametes are always haploid. So here I am adding gamete formation required meiosis. So chromosome become half. What we call gametes are haploid. They are having half number of chromosome. So what I am showing you, here there are pair of genes. When they will form gametes, they both will segregate. So I will make a square now. I will separate the gametes like this of this parent, and for female parent I will write the gametes here. Fine. Now what the gametes will do? Gametes will undergo fertilization. During fertilization, both the gametes will fuse. And when fertilization occur, capital TT and capital T small t and capital T small t will come. Here also capital T small t will come. So all four, they are having heterozygous combination. See, capital T small t, capital T small t, all four are having capital T small t. But what I told you, capital T is the dominant form. It will not allow the small t to express itself. So in this generation, this is called the progeny, the first generation, F1, first filial generation. In first filial generation, all were tall, or we say 100% tall plants we got. And what type of tall plants? Heterozygous. They were not pure, they were heterozygous tall. And on this basis, Mendel concluded his first law. And first law was law of dominance. On the basis of monohybrid cross, Mendel concluded law of dominance. What does dominance law say? When we cross between pure parents, like here we have taken the example of tall and dwarf, what we got? In F1 generation, only dominant. What was dominant? Tall trait was observed, while the another trait remains unexpressed. This is called recessive trait. So law of dominance says that when we cross between pure parents in F1 generation, only the dominant trait will express itself. I can show you this with one more example. Like if Mendel has selected not that height character, he must have selected some flower color. So the parent plant he selected, one was having violet flower, another was having white flower. This was male and this was female. I have written this as phenotype. If I write genotype, it will be capital VV. Genotype of this will be small vv. When they both will cross, the gametes will separate and we will get 4 different plants with the violet flowers only, right? So in F1 generation, we will get 100% violet flowers. But what type of violet flowers? Heterozygous violet flowers. This is called, again, law of dominance. Only the dominant trait will express itself in the F1 generation. This is the first law. This is the first law of genetics. First law. Okay. Now after that, okay, Mendel has also proceeded this first monohybrid cross. Let me show you how he continued this monohybrid cross to give his second law of genetics. For his second law of genetics, Mendel has continued his F1 generation plants as the parent plant, and he used them as parent plant, and then he got the second generation, and in second generation he find out some different result, and on that basis he has given the second law of genetics. Let me show you that. Okay, let's clean it first. Okay, let's continue here. So in monohybrid cross, when Mendel got his F1 generation plants, like if we have talked about the height, the F1 generation plants were capital T, small t, they all were tall. What type of tall? Heterozygous tall. So what Mendel has done, Mendel has self-crossed these F1 generation plants. And on doing self-cross of these F1 generation plant, what he said, the male and the female plant, they both were having same type of genotype. They both were heterozygous tall. And when they form the gamete, like this, they form the gamete. So here, the F2 generation was something different. It was not same as earlier. Again, there were 4 members formed, but all 4 were having some different genotype and phenotype too. Like here you can see one is having capital TT, and as you can see here 2 are having this capital T small t, and the last one, it is having small t only. So if I write them separately, the one is capital T, 2 are capital T small t, and one is small t, right? This is the same thing what I have written here. So this 1:2:1 is called to be the genotypic ratio. Genotypic ratio, because one was homozygous tall, 2 were heterozygous tall, one was homozygous recessive. Recessives are always expressed when they are homozygous. So this was the genotype ratio. And if I see them phenotypically, all 3, they were tall. So if I say the phenotype ratio the phenotype ratio was 3:1. Right now? When we self-cross the F1 generation plant, we get in F2 generation, phenotypic ratio is 3:1, genotypic ratio is 1:2:1. And why is it so? Because again, in F1 generation cross, the parent plants, they form gametes, and during gamete formation their alleles get segregated, separated, and when alleles get segregated, this gives them the separate ratio, the different ratio. The genotype ratio is different and the phenotype ratio is different, and that is the basis of the second law of genetics, law of segregation. Law of segregation was actually the extension of first law. When Mendel self-crossed the F1 generation plant, he find out that in F2 generation, the genotype and the phenotype ratio become segregate because of segregation of alleles during gamete formation. And that gives the second law of genetics. Okay. Then here comes the dihybrid cross. The word di is for two when two characters are studied at the same time. This is called dihybrid cross. So Mendel was fortunate that he has selected such type of characters that the both characters were related with seed either, or it is related with the pod either, it is related with the flower color and the position. All those characters were linked together. And that's why he find out some interesting result. What result he find out? Let's see. This is a seed which is round in shape and yellow in color. The another seed he selected was wrinkled in shape and green in color. So this was the parent generation. In parent generation, the phenotype was round yellow, wrinkled green. Genotype of round yellow was capital YY, capital RR, wrinkled green, small yy, small rr. Okay. Now when they were crossed together, in the F1 generation, they got all the seeds round yellow. Phenotypically, again which law? Law of dominance. In F1 generation, which law will be followed? Law of dominance always. Either it is monohybrid cross or it is dihybrid cross, always law of dominance will be followed. F1 generation was round yellow. They both were heterozygous. Now when he self-crossed these round yellow parent plants, he used this F1 generation as the parent generation and he crossed between them. Okay. So if this capital R, capital Y, small y, this was the round yellow Mendel get in F1 generation. With this round yellow, 4 types of gametes were formed. Again I repeat, when gametes are formed, all the alleles are free. They become independent. They can go, they can pair with any other allele available. So every allele will become independent. Capital R can go with capital Y. Capital R can go with small y too. Small r can go with capital Y. Small r can go with small y too. Okay. So both the parents will form 4 types of gametes. First gamete capital RY, second gamete capital R small y, third gamete small r capital Y, and the fourth one small r small y. Here we have these gametes. Look. From one parent 4 gametes, from another parent again 4 gametes, and the combinations which we got, we get like, see, I hope you all have played House Z. So here we have a line and we have this line. They both were, these both lines are showing yellow, yeah, round yellow seeds. Then we have this diagonal which is again showing round yellow. So how many are there? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 were round yellow. Fine. Then 1 round green, 1 round green, 3 round green. So 3 were round, shape was round, but the color was green. And the remaining 3 wrinkled yellow, wrinkled yellow, wrinkled yellow. 3 were wrinkled yellow. And this last one, the smallest one, only single, this was wrinkled green. So what was the ratio now? The phenotype ratio was 9 is to 3 is to 3 is to 1. When Mendel used two traits to be studied at the same time, which was considered to be the dihybrid cross, Mendel got law of dominance in the F1 generation, where all the seeds were round yellow. When he self-crossed these round yellow seeds, what he got? He got that in on self-crossing the F1 generation parent plants, he get the result of phenotype ratio as 9 is to 3 is to 3 is to 1. Should we repeat it once again? So, in the Mendel's law, 3 laws are there now. Sorry. First, second, and third. The first two laws were based on mono-hybrid cross. What was the first law? First law was law of dominance. What was the second law? Second law was law of segregation. And third law was law of independent assortment. This is the third law. And third law was based on, this was based on dihybrid cross. Fine. In law of dominance, F1 generation shows only dominant traits phenotypically. 100% result was there, 100% phenotype was dominant, right? They were heterozygous but in dominant only expressed. Law of segregation, F1 generation self-cross. F1 generation self-cross results into 3:1 phenotype ratio and 1:2:1 genotype ratio. Where? In F2 generation. When we self-cross F1 generation plant in F2 generation, the phenotype ratio becomes 3:1, genotype ratio becomes 1:2:1. And in law of independent assortment, in the law of dihybrid cross, we say when two characters, two traits studied in F2 generation during selfing of F1 cross, F1 generation, selfing of F1 generation, all alleles during gamete formation, assorts, arrange independently. And what was the result of this independent assortment? The phenotype ratio becomes 9:3:3:1. This is the summary. So these are the 3 laws of genetics based on monohybrid cross and based on dihybrid cross. We have to understand this thing that Mendel worked on the garden pea plant where he selected the pure line parents as the parent generation. In F1 generation of monohybrid and dihybrid cross, law of dominance was followed. When he self-cross F1 generation, in F2 generation, either the law of segregation was followed because of segregation of alleles during gamete formation, or in dihybrid cross, all the alleles independently assort them to give new combinations. The combinations which were present in parent, like see, here we were only having yellow, round and wrinkled green parents. But when we find out the ratio, in F2 generation, these two were totally different from the parent. They are showing variations from parents. And why they are formed? Because of independent assortment of alleles. Here you can see the round green, the capital R comes with small y. And here wrinkled yellow. Wrinkled yellow means capital Y comes with small r. Okay. So as I told you, in the F2 generation of dihybrid cross, the new variations are formed, and these variations were because of independent assortment of the alleles. Now after this, we will talk about some different topic other than Mendel's experiment. Here there was some exception. Mendel was not knowing about that exception. We will talk about the human blood groups. I hope you all must be remembering your blood group, right? In human beings, there are four types of blood groups. The blood groups in human, they are A, B, AB and O. Four type of blood groups are there. Yes, I know you all must having the query that ma'am we have O positive, B positive, A negative. These type of blood groups are existing. Yes. So that positive and negative is some other factor. This is known as to be the RH factor. So here RH stands for rhesus. Rhesus is the name of a monkey in which that factor, that gene was identified first. So in human blood cells, if this RH factor is present, we call it to be the positive group. And if this RH factor is absent, we call them to be the negative group. Okay. Let me tell you why this A, B, AB and O blood groups are there. When we say that blood group, so first of all, we are talking about the blood. We are talking about the blood cell, and in blood cell also we are talking about RBC. Fine. RBC is without nucleus cell in the body. It is without nucleus. That's why RBC is never regenerated. It is always replaced by the new RBC after 120 days. That is some different thing. Right now we will talk about the RBC. This RBC red blood cell is having the plasma membrane around it. On this plasma membrane, there are some antigenic factors or protein factors. We call them antigens. Basically, this word antigen is made up of anti plus gen. Anti is for antibody and gen is for generator. Any factor which can generate antibodies in our body is called antigen. It is basically proteinaceous in nature. So on the plasma membrane of RBC, there are specific antigen. If it is, suppose I am making it round, I call it A. Fine. In some other RBC, I make it triangular and I called it B. A antigen, B antigen. In some RBCs, here we have both the antigens. Like we will have this round one also and we will have this triangular one also. So what you will say? Both the antigens are there, A and B. Fine. And the last condition where on the RBC membrane there is no antigen, 0. So you will call it 0. Four types of blood groups are there. If the RBC is having antigen A on the membrane, A blood group. If the RBC is having antigen B on the membrane, B blood group. If the RBC is having both the antigens, AB blood group, and if the RBC is not having any antigen on the membrane, O blood group, O. Positive, negative. So if I write RH also here, RH also, so it will become B positive. If there is no RH, it will become A negative, like this. We have 4 types of blood group. How these blood groups are determined? How these antigens are formed on the plasma membrane? Again, there are some genes responsible. There are some factors responsible. These factors are existing in 3 contrasting form. Till now what we have said, every gene is having 2 contrasting form. But here is the exception. In case of blood groups, this antigenic genes are having 3 allelic form. And that's why we include blood group as multiple alleles example. There are not 2. There are 3 alleles in this. What are these 3 alleles? The gene which is forming A antigen, capital IA. The gene which is forming B antigen, capital IB. The gene is which forming no antigen. It is called to be the small I, O. Right? So there are 3 antigens for making there are 3 genes for making the antigens. And on the basis of these 3 alleles, we will get 6 genotypes. There are 6 genotypes. Like here, you can see IA with small I, IAA, IB with small I, that means 0, and these 6 genotypes are possible. So when IA comes with IA, the blood group will be, blood group will be A. When IA comes with the recessive form, then also the blood group will be A. When IB comes with IB form, blood group will be B. When IB comes with recessive form, again the blood group will be B. When IA comes with IB, both are dominant. This is forming the blood group AB. Here, very important point. This AB blood group is showing the example of co-dominance. Co means together. Both the dominant alleles are coming together. IA and IB. No one will feel inferior. Everyone will express itself and the blood group will become AB blood group. So this shows codominance. AB blood group is example of codominance. And the last one, when IO comes with IO, this is the only condition when the person will having the O blood group. So there are 6 possible genotypes responsible for all types of blood groups humans have. Right? So this is another important topic related with genetics. And the last one is the sex determination in human beings. We all know human beings are unisexual. The male and the female bodies are different from one another. The male body develops the male reproductive organ. The female body develops the female reproductive organ. How it comes so? How it happens? This is known as sex determination. So first of all, we have to understand that in every human cell, there are 46 chromosomes. As I told you earlier also, chromosomes always exist in pair. So 46 chromosomes exist in 23 pairs. Out of these 23 pairs, 22 pairs are exactly similar in male and female. Male and female. But the last pair, the last 23rd pair is different. In male, this pair is XY. In female, this pair is XX. Okay? So, here, in these pairs which are all same, we call them autosomes. And the last pair which is different in male and female, we call them sex chromosome. Right? And here you all can see the female are homozygous, both chromosomes are XX. The males are heterozygous, the male is having one X and one Y chromosome. Okay? Now, how they determine the sex? This is clear now that what is the arrangement of chromosome. What happened? When a male and a female undergoes mating or undergoes reproduction, the male body will form the, like male body is having the XY chromosome, the female body is having the XX chromosome, right? So this is the parent generation. Parent generation will form gametes. As I told you, in gametes we will make the box. Okay, it is overlapped. Just a minute. So here both the gametes, the male gamete we will write XY, female gamete I will write XX, and then I will make fertilization. So here will be XX, XY, XX, XY. What I can conclude from this chart? I can conclude that there are 50% chances that the baby will be boy and the 50% chances that the baby will be girl. Okay? So in human beings, what determines the sex? Chromosomes. Chromosomes determine the sex in human beings. This is called chromosomal determination. Chromosomal determination. But in some organisms, chromosomes are not determining the sex. Like in turtles, like in lizard, there are some examples of animals where environmental factors, specifically temperature, is responsible for determination of sex. High temperature can develop females, low temperature can develop males. So the gender or the sex of the organism depends on the environmental factor in some organisms. But in human beings, chromosomes are responsible for this sex determination. Again, this picture is showing you the same thing. The male and the female, the male is having XY, the female is having XX. So there are 50% chances of baby girl and 50% chances of baby boy. Why it is important to know? That because in India, female feticide is a very big problem. Females are blamed to give birth to girl child. But now you all understand this thing. It is not in hand of father or it is not in hand of mother, male or female, that what will be the gender or what will be the sex of the baby. It totally depends on the fusion of chromosomes. Which chromosome is going to fuse with what? If X chromosome of female is going to combine with the X chromosome of male, definitely the baby will be a girl child. But if the female X chromosome did fuse with the male Y chromosome, then the baby will be boy. So no one is responsible for the gender of the baby. This knowledge can control the female feticide problem in India. With that, I'm signing off for today. This is what we have learned about heredity. I hope you all have liked it. Go through that PPT again and keep learning. Thank you.